Hello everyone, I am Professor Charu from Ramjas College, University of Delhi, and we come to the end of our biotechnology series, basically animal biotechnology that we have talked about. The last aspect of this biotechnology series is that we talk about the gene therapy. So, we have talked about the recombinant DNA technology, we have talked about the various technologies that were developed based on it or in addition to it, such as CRISPR-Cas technologies and various different methods of gene editing. So, we have discussed all about them, and we also talked about the applications of this biotechnology in agriculture, in medicine. The last point is that we use it for the gene therapy. Now, to understand about the gene therapy, what is it, gene delivery tools, challenges and approaches to gene therapy forms the learning objective of this particular session. Now, what is gene therapy? Now, gene therapy, very crudely speaking, is basically a way to fix a genetic problem at its source, usually by adding a corrected copy of a defective gene. So, when we see that many diseases or many genetic disorders that we have discussed can be due to a particular gene. Now, when that gene is not functioning properly, it is defective gene, then that gene can be corrected or by adding a corrected copy, so that the functioning is restored and the disorder symptoms are reduced or removed. That is what is gene therapy is. Now, most commonly gene therapy uses a vector, which is typically a virus, to deliver a gene to the cells where it is needed. Now, this is in vivo gene therapy. We will talk about the ex vivo and in vivo in some time. Now, one important aspect is that gene therapy involves everything that we have discussed in our previous lectures. So, we talked about the generation of recombinant molecules. We talked about the synthesis also, even the corrected genes can be artificially synthesized as well. So, we have talked about the gene editing aspects. We'll see that gene therapy also involves not only adding the correction of a gene, but also repairing of a certain gene or silencing of a certain gene. So, in molecular biology, we have talked about regulatory RNAs, where we again discussed about the gene silencing as well. So, when you come to see this particular lecture of gene therapy, you should be aware of the previous things that we have discussed, because this is basically the application of all those techniques and technologies that we have discussed earlier. So, when we talk about vector here, we had I think 3-4 lectures on cloning vectors that we had discussed. So, these vectors are molecules which basically carry the foreign gene into an organism. So, for gene therapy, particularly the viruses are used, but for other purposes we have talked about various types of vectors such as plasmids, cosmids, BACs, PACs, YACs in detail, the characteristic features of all these vectors. So, in gene therapy, particularly viruses are used to deliver these to the cells. Now, to deliver a gene to the cells, we have already again talked about the different mechanisms of the gene delivery. We have talked about if it needs to be done, very small plasmid molecules needs to be introduced into another host organism, methods such as transformation, conjugation in prokaryotic cells can be done. But when we talk about gene therapy in humans or human cells, then basically we talk about the eukaryotic cells. So, again we talked about various delivery methods such as that gene gun method, microballistic method, or direct gene transfer method. So, we have talked about again in detail about all those things. So, gene therapy, we are talking in terms of application, the tools and techniques we have already discussed in our previous lectures. So, as I said, there are basically two types of gene therapy that we deal with. One is an ex vivo gene therapy and in vivo gene therapy. In vivo gene therapy is where we basically introduce the corrected gene into the vector by the process of recombinant DNA technology. We purify it and we inject it directly into the human individual. Now, obviously, there would be limitations, challenges and advantages of both the methods, but this basically is a direct method where we take the viral vector and we introduce this viral vector carrying the corrected gene directly into the human body. But what happened in ex vivo is that we isolate certain cells from the human, and in the petri dishes we introduce the viral vector containing our corrected gene, and then we grow them, and then we introduce these modified cells into the human body. So that is the ex vivo thing. Again, we are doing it in the living organisms cells only, but we are introducing it. So as you can see, just by telling the mechanism how it is done, there are certain challenges that will be faced or certain disadvantages and advantages faced for both the methods which is there. So where do we use in vivo and ex vivo? So ex vivo gene therapy approaches are generally used for conditions that are affecting the tissues that are either accessible and are easy to get in and out of the body, such as the blood and the skin. So if we talk about leukemias, the lymphomas or the leukemia, the blood cancer cells, so what we can do is basically we can isolate the cells, we can introduce the corrected genes, and we can put back the cells in. So whenever there is an accessibility or the ease to get in and get out of the cells from the body, the ex vivo gene therapy approaches are used. The cells, which are generally the stem cells, are taken from the patient, genetically manipulated and introduced back into the patient. So this is where the ex vivo gene therapy is used. In vivo gene therapies are the preferred option for organs that cannot be easily accessed or removed, such as, for example, you want to introduce the gene therapy to eye or brain or liver. You cannot isolate the liver, take it out from the body and you can put it back. So there what we do is, what is done is the scientists introduce directly the viral vectors containing the corrected genes into these organs. The vectors carry the neogenetic material or genome editing molecules into the patient's body, often as an infusion. So they infuse, they can inject, they can infuse, they basically introduce them into these particular organs containing the in vivo gene therapies. Now to be successful, a vector must, now when we talk about this vector in the in vivo gene therapy, the important characteristics what the vector must possess is that it target the right cells. Now these things basically limit the use of various different techniques and methods and give rise to the risks associated or the side effects associated with these biotechnology techniques when applied to human individuals. That one thing is that when a vector is introduced or infused into a particular organ, then they do not target the right cells. So it is very, very important that the vector know its destination. So they target the right cells. Second thing is they integrate the gene in the cells. The gene need to be integrated and become a part of the organism so that can be expressed continually and release functional proteins so that basically the disorder can be corrected. So integration the gene is very important. The third is the activation of the gene, which basically means that it does not just need to integrate into the genome and lie there without being functional. So whatever is needed, that also things or should be provided, or it should made use of the host organism's genome, etc. But the gene needs to be activated. Activated means it needs to be functional. And it avoid the harmful side effects, that is, it does not mount an immune response against it. Whenever anything is introduced into the body from outside, our defense mechanism, the immune response is basically works against it. So whatever vector you are introducing, it must have certain mechanisms or pathways or certain features in place so that it can avoid the harmful side effects which are there. There will not be a 0% side effect or 100% efficiency of the methods, and that is why the people keep on optimizing. That is one of the basically challenge for gene therapy, that we do not see that gene therapy is done at a very wider and elaborate scale, in which one of the factors is because of the side effects or risks associated with that it is found. But whatever, to be successful, these four things, the target right cells, integrate the gene in the cells, activate the genes and avoid harmful side effects is very, very essential. This is the vector toolbox. I will not go into much detail about them because we have talked in detail about the different kinds of vectors used in recombinant DNA technology to introduce the recombinant DNA into host cells. But what I just show here is that in gene therapy for human individuals, basically the viral vectors are used. So, the retroviruses are used, the adenoviruses are used, the adeno-associated viruses are used, or even herpes simplex viruses are used. Obviously, their virality, their virulence is nullified, but they act as the carrier for the foreign gene. The non-viral methods are also there, such as the use of the liposomes or even the use of the naked DNA. So, the naked recombinant DNA carrying the corrected gene can also be introduced into the cells for the gene therapy. So, there are many different vectors that can be utilized. Now, as I said, we need to address the various challenges that occur in gene therapy. The first and foremost challenge in gene therapy is the gene delivery and activation. Now, whatever viral vectors we are talking about or whatever methods we are talking about to introduce this, it's a very, very challenging process. The gene delivery method varies from cells to cells. It can vary from, what are our targets. It can vary where we need to target them. So, there can be, differences, and again, as I said, the integration is not the end point. Activation needs to be there. So, this gene delivery and activation needs to be optimized thoroughly so that gene therapy is effective. The second challenge which the doctors face is the immune response. As I said, our defense mechanism, our immune response mechanism is very, very, strong, particularly in, individuals, in all the individuals basically who are maintaining a proper health. So, if you are introducing this external recombinant DNA into the human body, it is going to elicit a immune response. So, sometimes even they use immune suppressants so that till the time the gene is delivered and activated, the immune system is suppressed a little bit by the use of immunosuppressants so that it does not reject the gene therapy molecules. The third is disrupting important genes in the target cells. So, this is again very important, that what happens is that there can be off-target activity also. So, whatever you are introducing, you want that particular gene only to be expressed, but it should not hamper the activation or expression of other genes in the cells. But sometimes what happens is that these vectors, when introduced, can disrupt the important gene in the target cells, leading to either the death of the cells or you are treating one disorder but you lead to another one. So, that can also be a very, very challenging aspect. The fourth aspect is again very important, which is the commercial viability. Now, what happens is that if you see then the genetic disorders, it will not be in many individuals. There will be a handful of population which is suffering from these deficiencies. But the production of recombinant human growth hormone, the optimization, everything requires a large amount of money. So, if the application or the commercial aspect is not that much and the production is too expensive, then the commercial viability concern occurs. And that is why not many companies produce these things, because they are not, there are a very limited consumers for this particular aspect. So, there are many challenges in gene therapy which needs to be addressed. Now, as I said, that it is not necessarily that gene therapy only involves the addition of a corrected gene. There are times when simply adding a working copy of the gene won't solve the problem. In such cases, preventing the cell from making protein the gene encodes, repairing the gene, or finding a workaround aimed at blocking or eliminating the protein is required. So, it is not that the gene is only defective, the gene might be working properly, but it may be not made properly, or the second thing is the gene will be defective because of certain sequential part that can be repaired, or sometimes what happens is that that particular gene is blocking or eliminating, or something else is blocking or eliminating the protein that it is going to play. So, in those cases the approaches differ. So, for example, the one thing is what is called as the dominant negative mutation. So, in dominant negative mutation, what happens is that the mutation occurs, and when the mutated protein is there, this mutated protein is going to interact with the normal protein and is going to hamper the work of that particular protein. So, it is not that there is not a functional protein which is there. There is a functional protein which is there, but the mutated protein is going to be dominant over the working normal protein, and therefore it is going to inhibit its particular activity. So, there is a corrected copy of the gene which is present. It is correctly expressed as well, but the mutated protein is dominant over this particular protein and therefore affecting its functional. Similarly, there can be gain of function mutations. So, what happens in gain of function mutations is that cell makes a protein that acts abnormally, causing problems all on its own. For example, it promotes abnormal interactions, it increases the interaction of the mutated proteins with its natural binders, and or promote misfolding or aggregation. So, it is not that there is a defective copy which is there, but by itself it is making an abnormal protein which is causing the defect in the particular deficiency of the particular hormone or whatever we are talking about, like growth hormone here. So, the gain of function mutation, these are some general mechanisms that I am talking about. They are not just affecting the production of growth hormone in a body, but these mechanisms can affect the production or expression of any gene in a particular body. So, there can be a gain of function mutation as well. Or the third aspect is there can be improper regulation. So, this improper regulation involve a protein that is functioning as it should, but there is a problem with where, when or how much the protein is being made. The genes need to be turned on at the right place, at the right time and to the right level. So, there has to be a very, very tight regulation, a very balancing that needs to occur. So, if there is an improper regulation, so the gene is perfect, so just the addition of a gene is not going to remove the negative effects of the disorder which is there, because everything is working properly, but the regulation is not working properly. The protein is formed, the protein is functional, the gene is working fine, but the regulation mechanism, the regulatory mechanism is defect based, so that it is not able to basically regulate the levels that what, where, when and how it is going to be produced. So, these are some mechanisms that require gene therapy approaches which is different from just adding a particular gene. And what are these approaches? For example, repairing mutations. So, there are many methods that are available. One of the method is, for example, SMART technology, which is the spliceosome mediated RNA transsplicing. So, what happens is it modifies the RNA. At the RNA level, it repairs the mutation that is occurring. So, when the gene is transcribed into RNA, then in RNA they make changes with the help of spliceosome, so that the transsplicing occur in a way that the RNA is basically in a correct form that is released. So, you are not adding any DNA, you are manipulating the already prepared RNA to become correct for that particular gene. Another mechanism is gene silencing that we have talked about. For example, RNA interference, triple helix forming oligonucleotide gene therapy, or ribozyme gene therapies. So what happens is these gene silencing mechanisms, and now the CRISPR-Cas editing also, it also creates gene silencing as one of the ways, or even in repairing, editing, all these mechanisms can be used, so that one can basically just not add a corrected DNA but repair the DNA which is there. Or if there is a regulatory mechanism which is going haywire, silence certain genes which are interfering in the production or expression of our gene of interest, for example, the human growth hormone. So that can also be done. Then there can be genetically modified immune cells as well. So nowadays what they are doing is they are producing the genetically modified immune cells, so that these immune cells are more resilient towards anything that is coming from outside, and therefore the gene therapy can work. Or by themselves they are carrying certain modifications or changes, that tools that is going to repair the particular gene disorder which is there. So these modified immune cells are also made and introduced into the body. Now, to end this, I just discussed certain success stories in India where the gene therapy is working, and in the recent 24-25 to 2024-25, this indigenous CAR cell therapy has been developed. CAR T cell therapy, which is basically the next CAR-19, it is called as. It is developed by IIT Bombay and Tata Memorial Hospital, and this involves the therapy for leukemia and lymphoma. So very promising therapy, the indigenous CAR T cell therapy. So what happens in this is that the T cells are modified. So the people who are having leukemia, the blood cells are removed, and from those blood cells basically the T cells are removed. So the blood is taken and the T cells are removed. Then the T cells are genetically modified to express a particular surface protein. Now this surface protein can recognize the mutated cells which are there, the tumor cells which are there. So these particular, you can see in the orange there is the T cells, and in the second step, that T cells are basically having certain surface receptors. So they are modified to produce these surface receptors, and these surface receptors have the more ability to combine with these cancerous cells much more. So they are proliferated, and then after proliferation they are basically introduced into the human body. Now these humans are basically sensitized by the use of chemotherapy as well. Before that conditioning is done for the person by chemotherapy, because this chemotherapy aids in the taking up of this T cells and as well as the functioning of the T cells. So this CAR T cell therapy has been developed and put in use by the Indian scientists which is working. So very, very important gene therapy and very useful gene therapy for the lymphoma and leukemia patients which is there. The next is the hemophilia A breakthrough. CMC Vellore scientists, they produce the gene therapy for the hemophilia. So hemophilia is basically, hemophilia A is caused by the absence of a clotting factor, which is the clotting factor 8. So what they do is, they, by the use of this gene therapy, they are basically repairing that gene which is there, and therefore the people, they do not release blood, they do not just bleed, which is the characteristic of the hemophilia A, that whenever a wound is cut, because there is a deficiency of the clotting factor, they can even bleed to death. So this hemophilia A breakthrough, the CMC Vellore scientists have developed this. So again, gene therapy is very useful, that is basically occurring. Then the spinal muscular atrophy. So this was a child that has been given the gene therapy for spinal muscular atrophy. And what happens is that they were again lentiviral vectors were introduced, but you can see the cost of this particular technology. It costed around 16 crore rupees for one individual, for one child, gene therapy to revert the spinal muscular atrophy Of course, she is doing well, she is obviously there are certain side effects which are there, but she is now standing, and she has started doing that because it was a couple of years back that they were given. But that 16 crore rupees, they raised this 16 crore rupees by crowdfunding. So crowdfunding is another important economic thing that can be used. You must have heard about crowdfunding, where the people put their problems out on the platforms which are dedicated to that, and then people donate the money for this thing. So they could even raise 16 crore rupees for the spinal muscular atrophy of this particular child. But just imagine that, how every person can make use of these technologies. So the commercial viability of gene therapy is a major cause of concern. With the advent of various new techniques and technologies, obviously the cost is coming down. Once we imagined that Human Genome Project took like 10 years and crores of rupees, and so many institutes were involved, and now you can get your genome sequenced in less than a week. And the cost is also not that much for a single sample. So that is how the biotechnology is blooming and giving rise to various techniques and technologies that are developing. So even we look forward that more and more advanced techniques and technologies will come, and the cost of the gene therapy will go down, because the gene therapy is a very, very promising thing. Obviously the optimization and lowering the side effects or targeting of, the off targeting has to be reduced. But again, it is a very, very promising technology that is developed. There are certain other examples of, for example, beta thalassemia treatment. The companies do it by lentiglobin gene therapy as well, or the sickle cell anemia treatment. So sickle cell anemia treatment recently, this Barsa 101, I think in February 2026 only, this has been CRISPR based gene therapy has been introduced for the sickle cell anemia treatment. So all these techniques and technologies which are involved in gene therapy are regularly advanced and optimized, and therefore gene therapy forms a very, very promising for all the for the treatment of the genetic disorders. And we look forward that the biotechnology becomes advanced in such a manner. But when I talked about the doping thing, I always want to emphasize on the ethical concerns and responsibilities of using a technology. Even the artificial intelligence is, aiding a lot in the development of biotechnology techniques, and therefore in gene therapy it is very, very promising. But the scientists, the people, the doctors, everybody has to use them in a very ethical and responsible manner, and not use it for any other purposes, for example, just to gain growth or mass or performance better and all those things. So biotechnology is definitely a very, very important thing for us, but it has to be balanced in the usage, and therefore it can treat so many genetic disorders which is there. So that's all from my side today. Thank you.